From viruses to whales, from fruit flies to dinosaurs, life comes in all shapes and sizes. What makes you, you? What makes an oak tree an oak tree? All living things inherit traits from their ancestors. That can mean physical features like eye color, or even behavior like a bird building a nest. For thousands of years, scientists had clues that traits were passed from parents to kids. They just didn't know how. That is until 1900 when a monk accidentally cracked the code of pea plants. Hi, I'm here to tell you the story of genetics. Let's start with the pea plant. Okay, not the most thrilling subject, but stick with me here. This monk/biologist was experimenting with pea plants because they have lots of traits that come in distinct flavors. Color, shape, size, texture, color. In scientific terms, these are called genes, like pea color. The gene controlling pea color comes in two flavors, green or yellow. Green is dominant, meaning if you have one green allele on one yellow allele, your peas will be green. Yellow is recessive. You need two yellow alleles to get yellow peas. Our buddy the monk discovered how these traits are transmitted by experimenting on pea plants, by crossing plants with different traits. He found that some traits blend, like mixing a yellow pea plant with a green pea plant results in green peas. Other traits seem to skip generations. After crossing purple pea plants with white pea plants, he noticed that some plants had purple flowers and others didn't. What was really fascinating was that some traits seemed to disappear, like white flowers. Then they would pop back up again in later generations. So what was happening? Why do traits sometimes seem to vanish? In order to figure this out, let's take a step back and talk about cells. Cells are the basic unit of life. Think of them like Legos. You put them together and bam, you get an organism. Every part of your body is made of cells, like skin cells, muscle cells, and brain cells. But there is one important thing about cells that you might not know. Every cell in your body contains identical DNA. Wait, what's DNA? Deoxyribonucleic acid, or DNA, is a molecule that carries genetic instructions. DNA is organized into structures called chromosomes. Chromosomes are like instruction manuals. They carry the genes that determine your traits. Humans typically have 23 pairs of chromosomes, one set from mom and one set from dad. During sexual reproduction, sex cells, sperm, and egg cells each have half a set of chromosomes. When a sperm fertilizes an egg, they combine their chromosomes. This means you end up with 46 chromosomes in every cell, 23 pairs. And guess what? All those chromosomes contain all the instructions needed to build a human being. So what happens when you cross two true breeding pea plants? Let's say you cross a pea plant with two dominant alleles for color versus another with two recessive ones. Each parent contributes one chromosome from each pair. That means each sex cell only has one chromosome with the gene for color. The sex cells split up and recombine randomly. Half of the sex cells will get the dominant allele and half will get the recessive allele. If a sex cell with the dominant allele fertilizes an egg with the recessive allele, the offspring will be a carrier of both the dominant and recessive alleles. And when it comes time to reproduce, that offspring will pass on one of those two alleles to the next generation. In this way, recessive traits can hide for a generation or two, but they never truly disappear. Voila, genetics. While the monk wasn't the first to discover this, he was the first to use math to describe it. He also had the right idea about how traits are transmitted, but he didn't know why. Enter Gregor Mendel, the man behind the laws of inheritance. His experiments were actually published in 1866, but they went largely unnoticed until 1900, around the same time scientists realized that chromosomes exist. It was then that the importance of Mendelian genetics was rediscovered. Scientists realized that genes come in pairs and are located on chromosomes, which are located in the nucleus of cells. And remember, genes carry the instructions for traits. Chromosomes carry the genes, cells carry the chromosomes. Wow! But what does DNA actually do? Well, here's the problem. Chromosomes and genes were identified in the 1860s, but we didn't know what they did until the 1940s. It wasn't until 1953 that scientists figured out the secret language of life, the discovery of DNA structure. Here's the deal. Everything in your body is made of chemicals. That includes your genes and chromosomes. So the big question was, what molecule was doing all that? Turns out it was DNA, and the discovery of its structure changed science forever. At the time, scientists knew DNA was a polymer, a string of smaller molecules linked together. But they didn't know what those smaller molecules looked like. Enter Rosalind Franklin and Maurice Wilkins. Using a technique called X-ray crystallography, they took pictures of purified DNA. Franklin's X-ray diffraction images showed that DNA had a helical shape. She also noticed that the helical structure was uniform throughout. That meant there were regular repeating patterns of atoms along the molecule. In 1953, Franklin and her team presented this research to the scientific community. Little did she know that two scientists at Cambridge University, James Watson and Francis Crick, used her data to build a model of DNA. They described DNA as a double helix, two strands twisting around each other. Crucially, they noted that the pairs of nucleotides on the rungs of the ladder faced inward. This gave them a pretty ingenious idea. Maybe the order of those chemicals spelled out the instructions of life. And just like that, the race was on. Watson and Crick raced against other teams to decipher the genetic code, but it wasn't easy. The chemicals in DNA don't align with the alphabet. There are 4 bases: adenine, guanine, cytosine, and thymine. How could 4 letters spell out the instructions for life? In 1961, a team of scientists solved this puzzle. They found that 3 letters of DNA code for one amino acid, the building blocks of proteins. Proteins carry out all sorts of functions in the body, from transporting oxygen to fighting disease, and sure enough, the genetic code worked. It was universal. The same genetic code was found in bacteria, viruses, plants, fungi, single-cell organisms, and multi-cell organisms. The genetic code is a universal language of life. In the mid-1970s, scientists began synthesizing DNA. By the late 1970s, scientists were able to cut DNA at specific points using restriction enzymes. Soon after, scientists developed polymerase chain reaction, or PCR, a technique to amplify DNA. Together, these techniques turned out to be incredibly powerful. Scientists could now copy specific genes, study them in depth, and modify them in various ways. By the early 1980s, scientists were creating recombinant DNA. They could take DNA from one organism and insert it into another. The first genetically modified organism, a bacterium that glowed green, was created in 1973. By the 1990s, genetic testing hit the mainstream. One example is the test for Huntington's disease, which was approved in 1993. Huntington's disease is a rare genetic disorder that causes the progressive breakdown of nerve cells in the brain. The disease is caused by a mutation in a single gene. At the time, there was no cure. The only treatment options were to manage the symptoms. But if you had a family history of the disease, you could now get tested. Of course, knowing you have a genetic mutation that leads to a debilitating disease is difficult. And at first, many people hesitated to get tested. Over time, though, genetic testing has become commonplace. Today, there are tests for many different conditions. Some predict the likelihood of getting a disease, while others diagnose it. Genetic testing allows us to understand our risks and make informed decisions. The Human Genome Project, an international collaboration launched in 1990, set out to map every gene on human chromosomes. This was a huge challenge, like finding a needle in a haystack. Haystack being DNA, needle being a gene. But in 2003, the project was completed and scientists had mapped the entire human genome. The ability to decode DNA ushered in a new era of science. No longer were we limited by what we could observe. Now we could study life on a molecular level. The year 2005 brought us the first DNA microarray, an incredible tool to study gene expression. DNA microarrays allow researchers to analyze thousands of genes at once. They can compare the activity of genes between samples, like healthy cells and diseased cells. This helps scientists learn how diseases develop and identify potential drug targets. The first DNA microarray was like flipping through a massive cookbook, comparing measurements of all the ingredients in a recipe. If you're making cookies, you may use flour, sugar, and chocolate chips. But if you're making pancakes, you may use flour, eggs, and syrup. Microarrays allowed scientists to view the entire cookbook, not just one recipe. In the 2010s, DNA sequencing became even faster and cheaper. In 2015, a group of researchers led by Stephen Quake at Stanford University developed a device that could sequence DNA faster than any other machine at the time. They called it the MiSeq. The MiSeq could sequence a human genome in just days. Compare that to the first human genome, which took over a decade. The pace of genetic research quickened, and we learned a lot more about ourselves. For example, we learned that humans don't have 23 pairs of chromosomes, at least not always. Sometimes we only have 22 pairs plus one. Instead of an X and a Y, we get two X's. People with two X chromosomes and one Y chromosome are much more likely to face fertility challenges. And today, gene editing is changing the world. With tools like CRISPR, scientists can cut and paste DNA. It's like having the ability to edit a text document. This opens up all sorts of possibilities. It may be possible to cure genetic diseases by removing harmful mutations. Researchers may also be able to enhance human traits by inserting beneficial genes. Take sickle cell disease, for example. This condition affects red blood cells. The cells become rigid and sickle-shaped, which can cause pain and organ damage. In 2017, doctors used CRISPR to remove the mutated gene responsible for sickle cell disease from a patient's blood stem cells. After editing the cells, they transplanted them back into the patient. And guess what? The procedure was a success. It was the first time CRISPR had been used to treat a genetic disease in humans. As we continue to explore gene editing, we'll face tough questions. Should we use CRISPR to eliminate genetic diseases? Should we use it to enhance human traits? Should we even mess with the building blocks of life? While we grapple with these questions, one thing is clear: the discovery of DNA changed science forever. It changed how we understand life. It changed how we treat diseases, and it's changing how we live our lives. And hey, if you're still watching, you probably love science as much as I do. So check out our other videos, all about amazing science stories that shaped our world. Thanks for watching.